Yeah, it's me. But it said, it seemed to think I was still on. Okay. Here we go again. It's recording still. I don't know. Doesn't matter because it's the same thing. Okay. There it is.
That's a blue dragon, right?
It is a blue dragon. Okay, so invertebrates make up 95% of all known animal species. I think on Monday I threw out the number. We have about 1.3 million known animal species and 95% of them are invertebrates. Okay, so this is the majority of animals. If I asked you to think of an animal, the chances are you probably think of a vertebrate and something cute and fuzzy maybe. Okay, but the majority of animals are not, I don't want to say they're not cute. They're just not fuzzy. No, I think these are adorable. This is not a fish because it's an invertebrate. Fish are vertebrates. So this has very few invertebrates on it. I don't have a lot of invertebrate clothing. I couldn't figure out what to do. It has an octopus. It has it has octopus. It has starfish on it, but that's all I've got. This is a blue dragon. It's called Glaucus atlanticus. It's a nudibranch, which falls into the mollusk phylum. They're pretty amazing little animals. If you've never looked up nudibranchs, that's something for you to do over break. There are a whole bunch of really crazy sea slug worm looking things that live in the ocean, obviously. These guys are pretty interesting because they eat Portuguese man of war, jellyfish. I don't know what you know about Portuguese man of war, but they're pretty big jellyfish. So when they eat them, they actually take their stinging cells from them and they concentrate them and store them in their body so they can use them to sting other organisms. So they basically steal the defense mechanism of another organism and make it their own. They also can take air in and hold it in their stomachs to keep them floating on the top, and they will sometimes group together into large rafts of these and wash up to shore and sting people. It's an invertebrate. It doesn't have a backbone. When we say invertebrate, that's not an official level of classification. That's not a kingdom invertebrata or anything like that. That's just a descriptive term for organisms that don't have a backbone. Maybe my browser is up to date. That's great. Could you just do what I wanted you to do? Why? There we go.
This must enter a sight. From the morning, or opening class up the lowers up front. And remember, we keep our Super 17 dangling to ourselves. That means you, Jimmy.
Has anyone yet memorized those? Okay, so they're not species. Mr. Ray is sometimes wrong. Those are not species, but those are phyla of invertebrates that he is mentioning in that song. And we're going to talk about most of them. Phyla and classes, some of them. So these are the pictures of the phyla that we're going to be covering. Again, this is not all of them. There are 5 questions you are going to think about while we talk about this, and you might want to even write these down, all 5 of them. Okay, as we're talking about each of these organisms, what you want to think about is, oh no, you don't want to think about that. How does it eat? So what is its feeding mechanism? They all feed differently, and that can be useful in classifying them. How does it exchange gases? How does it get oxygen and get rid of carbon dioxide? What is its mechanism? Does it have organs that do that, or how is it able to do that? How does it get rid of waste? Okay, that's one of the things that living things have to do. Everything makes waste, and so you have to get rid of it. Well, you'll get to the point in the semester where you talk about like how we do that, but every organism has to do that. I don't think we'll use the word poop for a long time. I probably won't even use the word poop. Can it move? If so, how does it move? Remember, we said most animals move, but not all animals move. So even though animal muscle tissue is unique to animals, not all animals have the ability to move. And then number 5, how does it reproduce? We're not going to go into detail for all of these. So that's kind of a question that if we talk about it, then you should know about it. But if we don't talk about it in class, you don't need to worry about it. Okay. So in your book, figure 33.3 gives you a good overview and description of these different phyla. It has pictures, and I don't know that my page numbers are right. I have 685 to 687. I don't know if that's right. This does not match with my book. So I'm assuming this might be right because it might be for the right edition. Okay, ready to go? With the first phyla. Periphery, and that's where we're gonna start. That's as far as I can get in the song, pretty much. Okay, so periphery are what? These are the sponges. As we said before, these are basal animals. Basal meaning they were the first to diverge from that common ancestor. So they're the closest to the root of the phylogenetic tree. We've already mentioned they're asymmetric. They're diploblastic, meaning they have what? 2 layers. They have an endoderm and an ectoderm, but they have no mesoderm. And we're going to throw in another fun vocabulary word. Oh, nope. They are sessile. Anyone know what sessile means? Same as sedentary. They don't move. Okay, so we've already answered one question. They don't move. So we have 5 questions, 1 down. Currently, all sponges are in one group, but that classification is under debate. Some people think they are monophyletic, meaning they all come from one group, and some people think they are not. Again, we're not going to have intense arguments about this in this class. You're not allowed to throw dots at each other if you feel strongly about this. Save that for another time. Okay, because they are the more simple organisms, again, we've already kind of mentioned this, they don't have true tissues, but they do have specialized cell types. And we're going to run out of, I'm going to write here and then I'm going to need this space, but we're going to do it anyway. So we're going to talk about some of their specialized cell types. So they have a structure to their body. It's not that they're structureless. They just don't have true tissues. So they have epidermis tissue. So that's what's on the outside, is the epidermis. And then between the epidermis and the inside layer, they have a layer called the mesohyl. And I was like, I've never heard of this. And then I found out it used to be called something else. And I was like, yeah, that's what it was when I was in college. And this is a gelatinous layer. It's a gelatinous layer of cells. And within that mesohyl, there's a specialized cells called Amiibo Sites. Those are inside the mesohyal. So yeah, they're in here. And Amoeba sites are kind of like amoebas, and you have a lab exam this week. How do amoebas move? Pseudopods. So these move like using pseudopodia too. So they can actually move within that layer. They can move around. And their function is to digest food. And because they can move, they can then transport those nutrients. So they can digest food and then help to transport those nutrients throughout the whole body. In addition to that, they can also, they're very multifunctional. They can also differentiate into other cell types. So they actually make spicules. Spicules are like crystal structures that provide support to the cell's body, the sponge's body. So they provide support and protection. And we will look at these when we get back from break. This is one of the things we'll look at as we'll look at a slide of the spicules. You can actually determine the species of a sponge based on what its spicules look like. By you, I mean people who study sponges.
So is it like a version of a cell wall?
Yeah, I mean, it's not a cell wall. It's extracellular, but it's like hard, like a cell wall would be. Okay, another kind of specialized cell that is going to under this white circle that I'm writing on now are Koanocytes or Choanocytes, however you like to say it, because people say it both ways apparently around the world. These are called color cells. That's an A, wow. They have flagella on them. So this is a choanocyte there. And then these blue ones that you're seeing are the Amoeba sites. And these yellow things are spicules. So choanocytes have flagella and they have a special collar around it. This, I should point up here. So you see the ball and then that little funnel shaped thing is the collar around the flagella. That helps to bring food in. So that's there to direct food inward. And they actually do phagocytosis to bring the food in. So they bring the food in, the flagella beat, that creates a current that helps suck the food into the funnel, the collar, and then the Amoeba sites help to break it down and transport it throughout the sponge's body. So it's a simple organism, but it does have a lot of specialized tissues. So how do they actually feed? First, you make an F. And then you make another F. They are filter feeders. Okay, so water flows in through these pores, which is how they get their name, porifera. They have pores throughout their body. Water flows in through there. The flagella then direct the food into the cells that are, and then all the rest of it flows out. So it's just a constant flow of water in through the pores and out through this opening, which is called the osculum. So water comes in through the pores and flows out through the osculum. And they just get the food out of that water. So obviously they're not eating large animals. Sharks are not eaten by sponges.
But that's what I was about to ask.
Yeah, so plankton, a lot of the plankton and protists that we talked about that live in the ocean could be their food. Diatoms, flagellates, that kind of thing. So we talked about how they eat. We talked about the fact that they don't really move. What else do we need to talk about? Waste. So waste and gas exchange. Oh my gosh, where's my finger? I need to sharpen my finger. Both waste removal and gas exchange happen via diffusion. So they don't have specialized tissues for getting rid of waste. They don't have specialized tissues for exchanging gases. All that just happens directly because the cells are in contact with water directly in and out of the cell membrane via diffusion. So it's all diffusion. So we talked about the fact that they can't move, but they actually can change their shape in relationship to environmental conditions. So they are responsive to the environment. So if water currents change, they will reorient their body to be able to get better water currents so they can get better feeding. So they're still responsive and not moving in like they're getting up and moving to a new place, but they're responsive to where they are and being able to respond to that. They can reproduce both asexually and sexually. So they can just do budding where a new sponge just grows off the side and becomes its own little sponge. So they can reproduce asexually by budding, but they do also reproduce sexually. They're what we call hermaphrodites. Do you know what a hermaphrodite is? Hermaphrodites mean that they are able to be both male and female. But in this case, they function, they're not male and female at the same time. They'll be male and then change to female, and so they can change, but they're sequentially hermaphroditic, not, I can't come up with the right word, not at the same time hermaphroditic. There's a better word for that I can't come up with at the moment. So they're sequentially hermaphroditic. Thank you. That's the word. I was like, there's some word for happening at the same time. It's called simultaneously. Yeah. So generally what happens is the sperm is released into the water and then it flows into another organism where fertilization happens. So it's not deliberate sexual reproduction. It's just release the sperm and the sperm go into another organism that's female and they make new little baby sponges. They actually do, when we say they can't move, they do have flagellated larvae. So after fertilization occurs and development begins, they have flagellated larvae. So those larvae can move and they will move around until they find, they want to get away from their parents because they don't want to compete with their parents. So the flagellated larvae are carried and have a little bit of control of where they go, and then they will attach to a surface and grow into a sessile adult. So when we say they don't move, we mean as adults they don't move. They do have flagellated larvae. And again, the advantage of having a flagellated larva is that the larvae get away from the parents and then they are not in competition with each other for food, which is the main resources that they would be in competition for each other.
Back to the whole hermaphroditic thing, are snails also?
Yes, there are a lot of invertebrates are hermaphroditic in different ways, and we'll talk about that.
So they're sessile until.
They're sessile as adults. Yeah, after the larval phase, once they've attached to some kind of substrate, then they develop into a sessile adult. So once they've attached, they're kind of done. They're not going to be like, oh, I don't really like it. We're going to pack up and move somewhere else.
What if they attach themselves to some sort of like sediment or like a bunch of gravel or something that can be uprooted?
And that's possible. They could attach to something that could move. But again, it's not them deliberately deciding, I'm going to move.
Yeah.
It's just they're the whim of whatever they have attached to. But yes, they can attach to things that move, definitely.
How long is the larva stage?
I don't know. I don't want to make up a number. A good generic teacher answer would be, it's probably different for every species. It's probably different for every species. And for some of these organisms, the larval phase is really long. So I don't know for sponges specifically, and it might depend on the species. Maybe dramatically even, I don't know. Yes. Is it what? Yeah. So these cells on the outside are just epidermal tissue. So that's just the outside layer. Then this is the mesohyl with the Amoeba sites and the spicules. And then these are the Koanocytes. They don't have enough differentiation to be considered true tissues in terms of function, I would say. Like even the Amoeba sites have multiple functions and can do different things. So they have a lot of jobs, so they haven't specialized enough to be considered true tissues, I guess is what we would say. Okay, last note of interest on sponges is some sponges actually produce antibiotic compounds. So that's something that people are researching. Can we get new antibiotics from sponges? We have a real problem where we have more and more bacteria that are resistant to antibiotics and less new ways to treat them. And so sponges might have some hope for us in developing new antibiotics, which I think is kind of wild. Oh my gosh, I have 7 more minutes, you guys. Oh, there's the spicule. See, all these are labeled probably. Yep, there's the mesohyl. There are the pores. There's the spongiceal. Oh, the spongiceal is the opening. I didn't say that.
That's
The opening on the inside of it is the spongiceal.
okay.
And the osculum is the opening. We talked about that. And there's a choanocyte, which I told you I was writing all over, and I did.
And you said the pores are where
That's where everything comes in, those purple openings. Okay, we're gonna start Naderi. So Naderi are our next phylum. So again, we're kind of working our way from the ones that diverged first, and we're continuing to get more and more complex. So starting here, everything has true tissues. So specialization to the point that the function is specific enough, it's considered a true tissue. Okay, we've already talked about things like these are radially symmetric, so these are still diploblastic. I don't know why I made it very big out there. And they're very similar to sponges in that things like gas exchange happen by diffusion. So both gas exchange and waste removal happen by diffusion. Again, these are organisms that all the cells are close enough to the environment that diffusion can happen between the organism and the environment. So they don't need specialized tissues to be able to get rid of waste or to exchange gases. This is the first level where we start to see some kind of nervous system, a very primitive nervous system. Called a nerve net because there's none of the cephalization, except we'll talk about the exception to that. There's not cephalization in that it's all the sensory information or something is concentrated at one end. The nerve net, it's spread out evenly throughout the organism. So it has the ability to like sense and respond to change in the environment, but it's not concentrated in one specific part of the body. It's getting that information from all around its body, which is true of a lot of radiosymmetric organisms. I had a friend in college who called them the nerve net. because it seems like sometimes his motor control is not centrally coordinated. I'm sure he'll be glad to know that I remember that and reference that. You can see here they have two different kinds of body plans. They can be polyps or they can be medusas. The polyp is the sessile form. So again, not moving. Some organisms only have a polyp form. So some Naderians only have a polyp form. The medusa is mobile. I think some organisms only have a medusa form, and some organisms in this phylum have both forms. So again, different kind of plans of attack about how to do life. Some of them sit back and let life come to them. Some of them move. When we say that they're mobile, some of them are mobile in that they are carried along by currents, and some of them have some control over that movement. If you've ever gone to an aquarium, and if you haven't, I'm sorry. Go to an aquarium that has a really good jellyfish tank, and you can just sit there and watch them all day. They're so peaceful. Just like, I could watch jellyfish all day. But again, some of them have no control over their movement. Some have some control over their movement. When we talk about polyps, ones that are always polyps, sorry, are Nemo's friends, the sea anemones, the Hydra, which we will look at in lab if they live. Whenever I get them, they don't live very long. So I'm hoping that they will arrive on Tuesday before Wednesday lab, and we'll have Hydra to look at because they're super fun to look at. And sometimes we can get them to eat, so we can feed them and watch them eat. And they're just adorable. Hydra are unusual because they live in freshwater. The majority of these live in saltwater. And then also coral fall into this category. So coral are Naderia, and we'll talk a little bit more about them. When we talk about medusas, we're talking about, whoa, that's not a J. The jellies. And really we should call them jellies and not jellyfish because they're not at all fish. Just like starfish are not at all stars. I mean, the way they are stars, they're not fish. I have been trying really hard to change my wording from starfish to sea star and have not yet made that transition successfully in my life. Jellyfish, I still call them jellyfish, even though again, nothing to do with fish. Okay, so what are our questions? How do they eat? We did not talk about that yet. Okay, so how do they eat? You can see here, they only have one opening. The mouth and the anus are the same. So everything comes in and then the waste goes out. But they're not eating in the same way that you and I are eating. So it's not as disgusting as you're thinking about right now. Okay, so they have a gastrovascular cavity. You can see in both forms, they have a mouth and anus as one opening. They have this gastrovascular cavity. And they can use their tentacles to bring food in both body plans. So definitely a more active eating plan than the sponges. Are they discerning? No. If they catch something, they're going to bring it in. So they're not necessarily like, oh, I don't think I would like that. I will not have that. They are carnivores. So they are only eating they're only eating meat. Again, they're not discerning in that something could accidentally get in there, but they try to eat meat. And then they just have enzymes in this cavity that break down the food. Making I think your book used the term like nutritious broth or something. And then it's, again, diffusion. It's just absorbed. Nutrients are absorbed into the gastrodermis, which is this inner layer. The lining of that gastrovascular cavity is called the gastrodermis, and nutrients are just dissolving into that. And again, because it's so small, they can just diffuse through the whole body.
When you say that carnivores, is that literally anything that's not a plant? Or
I would say yes. And again, I don't think they're super discerning, so pretty much anything. Okay, sorry. I didn't get to read you from my book about jellyfish, so that will be on Monday. Friday, today's not Friday. I will see you on Friday. And some of you I will see this afternoon. And some of you I will see tomorrow afternoon for your lab exam.